If you could shrink down and look inside any one of your cells, you'd find this bustling metropolis just humming with activity. And the workers, the machines doing all the work, they're called proteins. They do literally everything, from digesting your lunch to fighting off viruses. But here's the amazing part. All of these incredible, complex machines are built from just 20 different little building blocks, the amino acids.
And that kind of brings up a fascinating question, doesn't it? Our cells are these brilliant little chemists, so why can't they just make all 20 of these parts from scratch? Why are we totally dependent on our food for some of them? Well, to figure that out, we're going to dive into the cell's brilliant and sometimes limited architectural strategies.
Okay, first up, we're going to see how cells are the ultimate recyclers. They are absolute masters of turning what looks like common metabolic leftovers into brand new, valuable components.
So the fundamental strategy here is it's elegantly simple. Instead of building every single thing from the ground up, the cell often just grabs a molecule that's already floating around from other processes, like making energy. And then it just gives it a little chemical makeover.
The most common trick in its playbook is this thing called transamination. You can think of it like a molecular shell game. It takes an amino group, that's the nitrogen part that officially makes an amino acid, and it just swaps it from one molecule onto a new one. It's a quick, efficient little transfer.
And this is a perfect example of how it works. So, in step 1, the cell starts with oxaloacetate, which is a real workhorse molecule from our day-to-day metabolic cycle. Then, step 2, it snags an amino group from a donor, in this case glutamate, an enzyme makes the swap, and boom, step 3, you have a brand new amino acid, aspartate. It is molecular efficiency at its absolute finest.
But simple swaps aren't always going to cut it. For some of the most complex amino acids, the cell needs to fire up an entire assembly line. This is where things get really spectacular.
Just look at the contrast here. On the left, you've got these humble starting materials, pretty simple carbon chains, nothing too fancy. But on the right, the final products, phenylalanine, tyrosine, and tryptophan. Look at those complex, stable ring structures. So the question is, how on earth does a cell get from point A to point B?
Well, it does it with one of the most impressive feats of molecular engineering in all of biology, the Shikimate pathway. And this isn't just one chemical reaction. Oh no, this is a full-blown molecular factory. It's got multiple workstations, each one run by a specific enzyme, all of them working towards one single goal, forging those incredible rings.
And we are not talking about a quick two-step process. Just to get to the central intermediate molecule, a thing called choresmate, this pathway involves more than 7 major, distinct chemical transformations. It's this intricate, energy-intensive dance that slowly bends and folds and tweaks those starting materials until that aromatic ring finally takes shape. And here it is, this is the punchline. We can't do this. Animals, humans included, we completely lack the genetic blueprints for the Shikimate pathway. We don't have the enzymes, we don't have the factory, none of it. And that is precisely why phenylalanine, tyrosine, and tryptophan are called essential amino acids. We have absolutely no choice but to get them from plants and bacteria who can run this factory. Now, what's really cool is that these same principles of molecular construction, building from scraps or using these super elaborate pathways, they don't just apply to proteins. They also apply to the very letters of our genetic code.
So when it's building the basis for DNA and RNA, the cell actually uses 2 completely different strategies. It's fascinating. For the smaller rings, the pyrimidines, that's C, T, and U, the cell builds the ring first and then attaches it to the sugar. But for the bigger ones, the purines, that's A and G, it does the complete opposite. It painstakingly builds the ring piece by piece right onto the sugar foundation.
Let's just walk through that pyrimidine strategy. The logic is just beautiful. Step 1, forge the core ring from some simple precursors. Step 2, take that finished ring and just pop it onto an activated sugar molecule. And finally, step 3, perform a few last-minute chemical tweaks to get the exact base you need. It's another gorgeous example of modular, efficient design inside the cell.
Okay, so let's zoom out. We've seen all these incredible details. What does all this molecular architecture really tell us about the big picture? All right, let's boil it all down. What have we learned? First, that life builds this staggering complexity from just a handful of simple parts lying around. Second, cells have this amazing toolkit of elegant strategies, from simple chemical swaps to these massive assembly lines to build exactly what they need. And maybe the most important takeaway of all, our own survival is fundamentally interconnected. Our own biochemistry relies on the molecular labor of plants and bacteria to supply us with the essential building blocks we just can't make ourselves.
And that leaves us with one final kind of mind-blowing thought. Every single meal we eat is literally a transfer of these prefabricated molecular parts. At the most basic chemical level, we are completely dependent on a planet-wide biological supply chain. And when you think about it like that, this deep molecular teamwork, what does that tell you about how interconnected all life on this planet truly is?